Okay, let's dive in and, maybe get ready to rethink a little bit about medical science because we're kicking this off with something pretty surprising.
Oh,
Did you know that the way domestic rats first ended up in labs? Well, it was almost accidental. They went from being, pests,
right. Nuisances scurrying around.
exactly, to becoming pioneers in the lab. It's quite a story.
It really is.
So today, that's what we're doing, a deep dive into how biomedical research is evolving. Specifically, how we're starting to move beyond the lab rat, our sort of longtime partner in discovery.
And what that actually means for human health going forward.
Right. And we're basing this exploration on a really great piece from the SciShow YouTube channel called Are We Done with Lab Rats? It raises some fascinating points.
It does. It frames the whole issue very clearly.
Because, for over a century, let's be honest, these animals have been the unsung heroes, really, behind so many breakthroughs.
Absolutely. Countless discoveries owe something to them.
But things are changing now. The whole conversation is shifting, and pretty significantly.
Hmm.
So we're going to unpack the why behind that shift and look at some of the, well, frankly, amazing alternatives that are popping up in science.
Yeah, some really cutting edge stuff.
Definitely. So, okay, let's start right at the beginning. How did rats specifically make that leap from, common pest to lab stable? What was the turning point?
Well, it's interesting because it happened faster than you might think. Back in the mid-1800s, people actually started breeding them, but more for like weird fur patterns or even for sport.
Ah,
So,
yeah, not exactly science yet.
not yet. But then you had these pioneers, people like J.M. Philippa around 1856, and then Henry H. Donaldson in the early 1900s. They saw the potential.
What potential exactly? What made rats stand out?
A few key things, really. They were relatively easy to handle. They reproduce incredibly quickly.
Right. Short life cycles are good for studies.
Exactly. And perhaps surprisingly, they share quite a bit of genetic and biological similarity with humans. All that made them, well, pretty ideal candidates for controlled research.
So it started almost by chance, but then became very deliberate. And we hear about standardized rat strains. That sounds crucial. How did that come about?
Yeah, that was the real game changer. So by 1909, researchers figured out how to create the first standardized rat strain through inbreeding, basically, breeding closely related animals together over many, many generations.
Okay, so you end up with rats that are genetically almost identical.
Precisely. And the power there wasn't just, having consistent test subjects, although that was huge. It meant you could isolate variables in experiments like never before.
So it really ramped up the scientific rigor.
Totally. It fundamentally changed how hypotheses could be tested. And today, we have over 500 specialized rat strains, each one bred for specific research needs.
500. Wow. Okay, so that standardization is key. And you mentioned the biological similarity. That's the core of it, right? For modeling human diseases.
Absolutely. I mean, obviously they look different, but biologically and genetically, lots of parallels, even down to developmental timelines.
What do you mean by that?
Well, you can sort of age map a rat's development onto ours, so you can see how a disease might progress or how a drug affects the body over time, but in a much shorter window than in humans.
Okay, that makes a lot of sense. It speeds things up.
Right. And the similarity allows us to, breed rats or even genetically engineer them to mimic human conditions, things like multiple sclerosis, hypertension, even Alzheimer's. We get these living models to study.
And science didn't stop there, did it? I read about humanized rats. That sounds intense. What does that involve?
It is pretty advanced stuff. It started back in the 1980s, this idea of creating rats that actually host human cells.
Human cells inside a rat.
Yeah. So maybe they have components of a human immune system or human liver cells or even patches of human skin grafted on.
And the point of that is?
The goal is to create a model that behaves even more like a human system, to get a closer approximation of our physiology, which is super useful for studying things like, say, HIV or how certain bacterial infections take hold in human tissues.
It's really incredible how sophisticated these animal models have become. But it feels like there's a but coming. Despite all this, we're at a turning point, right? There are limitations.
There are, definitely. And it raises that really critical question. How well do these models actually predict what will happen in humans, especially with really complex diseases?
And the answer isn't always great.
Sometimes it's quite sobering. There was a big study in 2014, for example, looking at Alzheimer's drugs.
Okay.
It found a 99.6% failure rate for drugs that looked promising in rats, but then failed when they got to human clinical trials.
99%. 1.6%. That's almost everything.
Exactly. So while rats can model aspects of a disease, they often just don't predict the human response to a drug accurately enough for these complex conditions.
That's a huge disconnect, that translational gap you mentioned. Wow. And okay, besides the science not always translating perfectly, there's the whole ethical side too, isn't there?
Absolutely. Ethical scrutiny around animal testing has grown significantly.
What are the main concerns people raise?
It really boils down to questions about animal suffering, whether animals can truly consent to be research subjects, and ensuring the most humane treatment possible. As our science gets better, our understanding of animal welfare improves too.
Right. We know more about their capacity for experience.
Exactly. And that growing ethical awareness, combined with those scientific limitations we just talked about, well, that's really fueled the search for alternatives, methods that can, reduce, refine, or ideally replace animal models altogether.
Which brings us to the really exciting part, these alternatives. Let's start with in silico models, computer simulations. For those of us not deep in computational biology, what are we talking about here?
Yeah, in silico just means done on computer. So these are basically sophisticated digital simulations of biological stuff.
How do they work? What do you feed the computer?
You feed the algorithms tons and tons of data, everything from molecular chemistry, genetic information, how drugs interact, clinical trial results, all of it. The computer then builds models to predict outcomes.
And how good are they? Can they really predict biological effects?
They're getting surprisingly good. For instance, some machine learning models can now test if a chemical is likely to be toxic with up to 78% accuracy, which is pretty impressive.
78%. It's quite high, but I guess they're not perfect. What are the downsides?
Right, there are still challenges. The big one is often called the black box problem.
The black box.
Yeah. The model might predict what will happen, like this drug will probably fail with decent accuracy, but it doesn't always tell you why.
Ah, okay. You lose the mechanism.
Exactly. And for a lot of biological research, understanding the why, the underlying process, is just as crucial as predicting the outcome. You need that insight to design better therapies next time.
That makes sense. It's a trade-off between prediction and deep understanding. Okay, so from simulating biology digitally, what about actually building tiny bits of human biology in the lab? That brings us to organoids.
Yes, organoids are really revolutionary. Think of them as 3D mini organs grown in a dish.
Mini organs. Seriously, how?
They're typically grown from stem cells or sometimes small tissue samples, and you can coax them to develop into structures that mimic the function and even some of the architecture of real human organs, like tiny little brains or livers or guts.
Wow, and the benefit there is you can study human tissue directly.
Precisely. Ethically, too. You can, say, infect mini-brain organoids with the Zika virus to see exactly how it damages human brain cells or study Alzheimer's plaques forming in neural tissue. All in a dish, no animals involved for that specific step.
That sounds incredibly powerful. But are they perfect replicas? What are the limitations there?
Good question. No, they're not perfect replicas. One issue can be inconsistency. Getting them to grow reliably the same way every time is still a challenge.
Okay.
And maybe more importantly, they lack the full body context. An organ in your body has blood flow, it interacts with the immune system, it gets signals from other organs. An organoid in a dish doesn't have all that complexity.
Right, it's isolated.
Yeah.
So how do we get closer to that real body complexity? I've heard about organs on chips. That sounds even more futuristic.
It does sound like sci-fi, but it's very real. These are basically tiny microfluidic devices, little chips, often clear plastic, that simulate specific functions of human organs.
How do they work?
They have tiny channels and chambers where you put living human cells. You can flow fluids through, kind of like artificial blood vessels, creating a microenvironment that mimics what happens inside the organ.
Okay, so it's less about structure and more about function.
Kind of, yeah. And they have some advantages over organoids. They tend to be more reproducible, you can scale them up more easily, and they're often more stable for longer-term experiments.
And the really cool part, I gather, is that you can connect these chips together.
Exactly. That's where it gets really powerful. You can link, say, a lung chip to a liver chip to a kidney chip.
To see how a drug affects the whole system.
Precisely. How it's metabolized in the liver, how it affects the lungs, how it's excreted by the kidneys. You can model those complex interactions between organs.
And this isn't just theory, right? People are actually using these. Oh, yeah. It's happening now. NASA is using them to study how spaceflight affects human physiology.
Right.
And maybe even more significantly, the FDA, the agency that approves drugs, is already starting to use data from organs on chips in their assessments.
The FDA, wow, that's a huge validation.
It absolutely is. It signals a real shift, a growing acceptance that these technologies can provide meaningful data, potentially reducing the reliance on traditional animal testing for some aspects of drug development.
So putting all these pieces together, the simulations, the organoids, the chips, what's the big picture for the future? Are there any truly moonshot projects happening? There are some really ambitious things underway. For example, I know researchers in Canada are working on building the world's largest computerized human brain.
Computerized brain.
Yeah, the goal is to simulate cognition, how we think, and how drugs might interact with the brain on an unprecedented scale. Imagine being able to test psychiatric drugs or dementia treatments virtually before they ever go near a person.
That's mind-blowing. It could change everything in neuroscience and drug discovery.
It really could. And alongside these huge simulation projects, we're also seeing more clever ways to do early studies in humans, ethically.
Like what?
Things like microdosing, giving people incredibly tiny doses of a drug way too small to have any therapeutic or harmful effect, just to see how the body processes it using super sensitive instruments.
So you get human data very early, very safely.
Exactly. That plus advanced imaging techniques lets us get insights directly from humans much earlier in the process. And again, regulatory bodies like the FDA are increasingly open to data from chips and organoids, which directly chips away at the need for some animal tests.
So it really feels like, as you listen to all this, we're in a major transitional period for biomedical research.
I think that's the perfect way to put it, a transitional era.
The lab rat isn't, gone overnight. It's not obsolete across the board.
No, it definitely still has roles, especially in certain types of basic research or complex whole organism studies. But it's clearly not the only option anymore, and maybe not always the default first choice.
Right. And for you listening, what does all this actually mean? Why should you care about moving beyond rats? Well, the goals here are pretty compelling. First, it's about improving the accuracy and, crucially, the relevance of the research, getting results that are more likely to actually translate to human health.
Better predictions, hopefully leading to better treatments.
Second, it tackles those ethical concerns head-on by minimizing the use of animals where possible, which aligns better with, evolving societal values.
A really important driver for many researchers and the public.
And third, think about personalization. If you can grow mini organs or use organ chips with your specific cells.
Right, patient-specific models.
Exactly. That brings us so much closer to truly personalized medicine, where treatments could be tested for you specifically based on your unique biology.
That's the long-term vision, and these technologies are paving the way.
It's an incredible prospect. So we've covered a lot, from the accidental history of lab rats to these really cutting-edge digital and biological tools.
Yeah, it's a huge shift.
Which leaves us with a final thought for you to chew on. As this research becomes more focused on human cells, human data, human simulations, what new ethical questions do you think might pop up?
That's a good one. Like with hyper-realistic brain simulations or highly personalized organoids, what new boundaries do we need to consider?
Exactly. How might all this advanced human-centric technology reshape our very understanding of what it means to study life itself? Something to think about.